Hey everybody, Dr. Viso here. In this video, we're going to talk about transplant rejection, and this is a pretty big video. A lot of topics integrated here, so we'll talk with just some basic principles of transplantations to start. We'll go through the different types of transplantations, talk a little bit about graft versus host disease, and then we'll recap this all kind of like we've been doing with some of the last chapters. And finally, we'll talk a little bit about some of the immunosuppressants that can be particularly high yield, as well as some of the biologic immunotherapy. Just to get things started, starting off pretty simple here, when we're talking about the principles of transplantation, all of these different terms have to do with where a donation is happening from. Okay, so let me show you what I mean. So in general, we have a donor. Okay, we have a donor and we have a recipient. And if you've been following along from the previous videos where we talked about red blood cell transfusions, for example, we talked about the same terminology, right? It's a donor and a recipient. The only difference was we were talking about transfusing blood or platelets. Here we're talking about transplanting an organ, essentially. And so a lot of the times we're going to talk about this transfer, whether it's a kidney or skin or whatever it is, we're going to say that it is a graft. Okay, so essentially the donor is going to be giving a graft to the recipient. Now, who the donor is, who the recipient is, that's where all of this terminology comes into play. So let me show you what I mean. So for example, let's just say that, we have a patient that has a really bad sacral wound ulcer, okay? And sometimes what we do is we would put a flap over that sacral ulcer, right? Because we don't want it to get infected. We don't want that patient to get osteomyelitis, right? So we can take skin from the same patient and put it over the ulcer. So what I'm essentially saying here is the same patient is the donor and the recipient. The patient is simply donating skin, maybe from their thigh or their leg, and that skin is then going to their sacral wound ulcer. Okay, so in this situation, the donor is the recipient. And so the term for that is an autologous transplant. Okay, so it's the donor is the recipient. So it's donor to self. And what I would call this type of graft, would be an autograft. And again, that means that the donor graft is going to themselves, maybe just a different part of the body potentially. All right, so the next situation, let's say that I have a patient that's donating a kidney to another patient. Okay, so here's my patient one. They're donating a kidney to another patient. Okay, so here's a different patient and they're donating their kidney, right? That's the graft. It's going to this patient. Okay, now because we have two different patients here, unless they're genetically identical, there's going to be some differences in their HLA antigens, which I'll talk about in a second. Okay, but the point is that these are two genetically distinct individuals. And so this type of transplantation is called an allogeneic transplantation. Okay, so that's an allogeneic transplantation. We're donating an organ in this situation to a non-genetically identical individual. All right, and so again, going with this prefix here, auto, right? Previously, this would be allo, right? This is an allograft. Okay, so this would be an allograft. Okay, so this is the type of graft would be an allograft when it goes from one person to another if they're not genetically identical. Now, in a situation if they were genetically identical, okay, that would be if you had, for example, two patients potentially that were twins, right? Particularly monozygotic twins, right? So if you had two patients that were monozygotic twins and you had one patient donate some tissue to another, then we would call this an isograft. Okay, iso meaning same. They're genetically the same. Okay, so that would be isotransplantation, donation to a genetically identical recipient. Now you're not going to see, the isograft mentioned quite as frequently because again, an isograft, it has to be between genetically identical individuals, and it's really only going to be the case in a board question where they're talking about monozygotic twins. So most commonly in a board question, you're talking about allografts. In general, when you talk about transplant rejection, you're not really going to be talking about rejection in these first two categories, right? So the autologous transplantation, the isotransplantation, there's not any differences in the genetics between these individuals. So we don't expect there to really be much rejection. Rejection comes because either the donor or the host is sensitized to some new antigens. Those antigens are going to be the HLA molecules on the cells that are being transplanted, or it could be on the host cells as well, which I'll talk about as we go through this. Now, the last one I just want to put on here before we wrap this up, because occasionally you'll see this come up. Let's just say that we're transferring tissue from a different species to a human. Okay, so to some patients. So this happens a lot when you talk about certain types of heart valve transplants that aren't necessarily allogeneic, right? They're not coming from human to human. It's coming from a different species, usually bovine heart valves you'll see occasionally. But when you see this terminology for the graft here would be a xenograft. Okay, so xenograft is when we're talking about donor grafts to a different species. Okay, so that's a xenograft. And again, the biggest one on here is going to be the allogeneic, right? The allogeneic is the big one because that's how the majority of these transplant rejections are going to occur. They're going to occur because there wasn't complete matching between the donor and the recipient. Okay, so this is the majority of what we're going to be talking about. Now, how do these antigens even cause a rejection to begin with? Well, remember, the HLA genes will code for our MHC molecules, okay? And we talked a little bit about MHC being on the surface of all nucleated cells, particularly MHC1, right? MHC1 is on the surface of all of our nucleated cells. And the thing that we talked about earlier too is MHC1 can be remembered by remembering that it's just one letter after the HLA molecule, whereas MHC2 can be remembered because there's two letters after the HLA molecule. And these are all different variants of these HLA molecules. And so there's a lot of diversity and distinction between them. The point being, though, let's just say that, we did donate some organ from one patient to another. There's a couple different things that can happen, right? So let's just say that this recipient here, right, this recipient might recognize MHC molecules on that transplanted kidney, for example, right? They might recognize MHC1 and say, hey, this MHC1 has a different HLA than the HLA that we have, right? I'm doing gross oversimplification here, but they might have slightly different HLA molecules. And so the T cells in this patient, particularly, right, what's going to bind MHC1? It's going to be CD8 positive T cells, right? Rule of eights. So the CD8 positive T cells in this patient could attack this graft. And it can attack this graft, this allograft, right? It can attack it at these HLA antigens, at the MHC1 antigens. Now, the other thing that can happen is we can also have antigen-presenting cells that say, hey, look, these HLA molecules that have the two letters after the HLA, these HLA antigens are different from our HLA antigens. Again, but this is now at MHC2, right? This is going to be antigen-presenting cells. And again, if you're not familiar with some of this, go back and watch some of the videos on T cells that we've talked about. We're really building on the knowledge from this whole video series to get through this concept. But the idea here is you can have antigen-presenting cells bind this MHC2, present this to CD4 positive T cells, right? CD4 positive T cells can then get activate B cells through CD40 ligand, and you can generate antibodies against these antigens, okay, via MHC2. Okay, so this could be potentially more of a humoral response. But we'll get into all of the details on this as we proceed, but that's the concept. That's essentially what we're talking about. So the big take-home points here, remember, allogeneic is what we're primarily talking about. Two genetically non-identical recipients, right? They're both humans, same species, that we're having one organ donated to another. If the patient was genetically identical, very unlikely for this to happen because they're going to have all of the same HLAs, right? So you're not really going to see a significant reaction there. And if it's autologous, it's going from the donor to self, essentially. So it's the same person. So generally, we're talking about allograft transplantations. And again, it could technically happen here with the xenografts as well. They just don't tend to come up as much in terms of the board questions.